All of the answers in the world are right there at your fingertips. People can't question anything anymore. And I get a bit emotional about this because it drives me crazy as a questioner. I'm a natural questioner. We need more innovators.
After a quarter century of working with private and corporate wildlife management programs across the country, I've seen what truly works and what doesn't. I'm Jason Snavely, certified wildlife biologist and private wildlife consultant. I'm sharing more than 25 years of experience through this podcast. My knowledge of wildlife ecology and management allows me to sort through the propaganda and report only the facts. My work places priority on the legacy of the land, the legacy of the private American landowner, and ultimately, the future of thriving wildlife populations and the habitats they rely on.
Nine most terrifying words in the English language are, "I'm from the government and I'm here to help."
Did you ever feel like you're only getting a fraction of the story when it comes to a particular situation or topic? Have you ever thought to yourself, there's got to be more to this story? I wonder if anyone else has a plethora of questions about this like I do. This is exactly how I've always felt about chronic wasting disease, and the truth is, I'm not alone. Many of my former wildlife professors and teachers and hundreds of my wildlife colleagues consider me a voice in the wilderness when it comes to CWD. I've always just kind of sat on the sideline and asked questions surrounding this whole debacle until now. In this podcast, I'd like to present the rest of the story. I'll dive deeper into the one generally accepted theory that so many of my colleagues hang their hat on, without questioning its origin, the data, and the story behind it all. This is the prion theory. While I'm not saying that a normal host protein is not involved in this disease at all, I'm asking the question, is a misfolded prion what a reputable scientist should be calling PRP amyloid really the infectious agent? Perhaps of more importance, whatever happened to the research looking at latent viruses and contaminated feed? After all, I don't think anyone is arguing that CWD can't spread through the environment. So let's talk about chronic wasting disease and why license buying sportsmen, career wildlife professionals, academicians, and nonprofit organizations are in a perpetual state of divergence without any real tangible scientific or management progress to speak of. In this podcast, I won't get into the millions of dollars spent on research, but truthfully, most of that research simply can't be justified from a return on investment standpoint. Many researchers and managers will argue with my point. However, if you challenge them to invest millions of their own declining and eroding U.S. fiat dollars to discover what we currently think we know about their prion diseases, their eagerness to fund said research would erode. Their financial support simply wouldn't exist. After all, it's always easier when it's someone else's money you're spending. I've been following the CWD story since 1997, 3 decades ago, as an undergraduate student of wildlife science at Mississippi State University. More recently, I've watched as some sportsmen finally ask better questions. Many frustrated whitetail fanatics have formed social media groups where they regularly voice their concerns with policies laid out by state agencies. And I think this is actually great. Most notably, hunters are frustrated with the reckless killing of non-symptomatic deer to slow the spread of a disease that they know so little about. They certainly don't have the peer-reviewed data that prove killing more deer saves more deer. The fact is, it's a bad time in history to pull the slow the spread card. Do these people even attempt to read the room? Perhaps they're tactical and they learned from the psychological warfare successfully employed during the COVID debacle. So many profited and enjoyed power and control over the obedient masses. It's a powerful tool to employ against those who struggle to think on their own and really just ask better questions. Over the years, I've rediscovered much of the ignored data and some of the lost research, and as a result, I too have many questions that I'd like to share with you. Unfortunately, we're losing facts by the day, but we still have plenty of data to prove that Prusner's science is not science at all. I hate to admit it, but even I have been surprised with just how messy and sloppy this debacle is. In the past, I've been known to simply question what we think we know about CWD and all transmissible spongiform encephalopathies. Until now, most of my input on CWD has been contained in private discussions with my clients and those colleagues who are able to have civil discussions without letting their emotions or their dependence on their salaries get the best of them. I openly wrote a few articles about CWD in my Whitetails column for Peterson's Bow Hunting, which turned out to be very popular pieces. Thankfully, my editor gave me the freedom to report the facts, but he still labeled me a voice in the wilderness. The truth is, if you go opposite the herd on any popular and emotional topic in life, notably if money and power are involved, you're not going to be very popular. I've also avoided CWD because it can be a very complex topic that so few understand, including the wildlife experts. The truth is, most of the good work on TSEs has been done by veterinarians, yet the lion's share of educational material, albeit inaccurate, constantly flows from employees of state wildlife agencies. Who are these people and who labeled them experts? None of them are reading the foundational papers, and none of them are conducting any infectivity investigations themselves. If you think wrapping your mind around the disease is difficult, try telling wildlife professionals about what you see in the lost and ignored literature and what you're seeing on the ground, where whitetails live, especially if it's the opposite of this sloppy narrative that we've all been force-fed. Don't ask my colleagues any questions that don't fit the prion narrative, or you're automatically lumped in with the CWD deniers. Another fun fact about representing a voice in the wilderness among your peers on a highly emotional and political topic is it's just not profitable or good for one's career to question the management of the most popular big game animal when it threatens the livelihood or financial gain of so many others. I decided to start talking about the questions that I have after decades of watching the situation unfold. I'm not looking to make enemies, but I also have enough friends. Instead, I'll simply represent the white-tailed deer and their cervid cousins from the perspective of doing what's best for the resource. The fact is, no one is asking relevant questions, and agencies are failing miserably with bad policies. I've been fortunate to get to know some truly talented scientists who were on the scene when the modern day story of Scrapie and CWD began. They were there to discover what was going on with Kuru amongst the Foray, a cannibalistic New Guinea tribe. They were privy to the scientific journey of Gibbs and Gajushek, who investigated whether ALS, multiple sclerosis, Alzheimer's, and more than 40 other neurological disorders might also prove infectious. They were there when one man and a select few of his loyal lab rats changed the TSE scene forever. I find it interesting that scientists have made very little progress in all of the aforementioned neurological diseases. Does that sound familiar as we discuss CWD? What are the holes in the science? And perhaps more importantly, what about the lost science? How come no one is talking about the prion story and the politics of the prion name game? Follow along because I will. But first, I'd like to lead off with a fun piece called CWD, The Rest of the Story. I'd enjoy hearing from each of you on this topic, so drop me an email, jason@droptinepodcast.com. Keep in mind, this is just a teaser to lead off my work on CWD. There's so much more to come. I recently created a podcast called Drop-Tine Podcast YouTube channel, where I expand on this work with more details on the gaps in the CWD debacle. My goal is to bring some great questions to light, and who knows, perhaps the talented scientists on the scene today, like Seabury, Henderson, and Haley, will actually look for other potential infectious agents. Make sure you subscribe to the Drop-Tine Podcast YouTube channel to see more. To introduce some of the lost facts and hidden details surrounding the popular prion hypothesis, I'd like to honor the late, great Paul Harvey while poking fun at human nature and really our ability to erase or perhaps ignore vital historical facts from important topics. So many of my wildlife professional colleagues don't read literature older than 5 years. That's a huge mistake. And it's just not good science. This piece and the rest of my CWD work is for those hunters and sportsmen who are collectively questioning the sloppy nature of how state agencies and wildlife professionals are mismanaging this disease. What do I mean by sloppy? You've all heard the Missouri experts refer to CWD as contagious. Anyone in medicine or biology knows this is false. It's false information. Do your job, do better. One of my major motivations to do this work was initiated by my attendance at a very poorly presented educational event on chronic wasting disease in Benton, Pennsylvania. As a wildlife biologist who's actually read the science, I couldn't believe one of my fellow wildlife biologists, a state agency biologist, was presenting an opinion piece on CWD, but selling it as scientific fact. The data were misrepresented to wrongfully call out deer breeders as a major cause of the spread of CWD. And even worse, we weren't allowed to openly ask the expert any questions after his presentation. More concerning to me was that the audience there to learn about CWD was consuming this misinformation without question. This piece is also in part for those who assumed that the book is closed on CWD, falling hook, line, and sinker for the prion hypothesis that came out of one man's lab, a hypothesis rooted in a scheme to merely win a prize and capture fame, and a hypothesis that resulted in over-interpreted data from findings that were not reproducible outside of his own lab. What does the Prusner prion cabal have to say about my work? They've been invited on the show, but they want nothing to do with it, like a few others. This work is in part supported by some amazing scientists, science writers, and investigative journalists who, from the past, unfortunately realize that their time is better spent elsewhere, considering the politics and the lack of funding for legitimate science. But mostly this work is supported by my family, who have sacrificed by me spending countless months and years following along, asking questions, and talking to the true experts from all corners of the globe, some of which don't want to be heard because so many of you don't want to listen. The unfortunate fact is, so many facts have died with talented investigators, while other valuable information rests in the minds of scientists who realized long ago that it's just not worth the fight anymore. And instead, they opt for a relaxing retirement. But most importantly, this work is for the white-tailed deer and the sportsmen and women who, like me, want better answers, as we realize that the resource is being depleted as the reaction to the disease is exponentially more deleterious than the disease itself. I've spent 25 years as a private wildlife biologist. I've worked with whitetails in every part of their range, and I've been fortunate to have the freedom to speak my mind. Supported by formal education in wildlife science, combined with those 25 years of working in the field with talented private land handlers. I'd also like my fellow sportsmen and private landowners to hear this work so they can individually determine if the management approaches employed by state agencies are justified. I'd like to provide some educational content that illustrates just how little the experts understand this disease. It's time we respectfully demand more for the resource that we as land handlers and hunters love so much. But I'd like to point out that it's crucial for us to fight this fight with facts, real data, and some of the much needed data are there. The rest has yet to be uncovered, as scientific investigators are chasing perhaps the wrong questions. Let's discuss it in a professional manner, but all questions are fair game. Before we rip, I'll lay out a brief summary of what the experts believe about CWD, just in case you've been living under a rock, or perhaps, like so many, you're just avoiding the topic because the scores of experts can't seem to agree on even the most elementary aspects of the disease. If you've heard or read it once, you've likely heard or read it dozens of times. We've been told to believe that CWD is an always fatal, hard to understand, and even harder to describe prion disease, a reportedly novel class of infectious diseases known as transmissible spongiform encephalopathies, or TSEs for short. In truth, the word transmissible is misleading from the very start. Infectious would be a much better word. They want you to believe that the science has proven reproducibly and beyond the shadow of a doubt that TSEs are not caused by a latent virus or a bacterium or even tainted food, but instead by a naturally occurring prion protein, or PRP, that becomes mysteriously misfolded PRP amyloid and resists being broken down by the body the way normal proteins are. One man created the narrative that this rogue protein that he believes lacks nucleic acid, or does he, can multiply. He invented a name for the rogue protein in a very unscientific process rooted more in melodious intent than in science. In fact, reputable scientists would have simply named this normal host protein GP34, a glycosylated small protein of 34 kilodaltons. I repeat, one man successfully sold his theory that a rogue protein, which he claims to have discovered, needed a new name, and then suggested that it may be the infectious agent, until he later redacted that statement to say that he never said that only a protein is involved. Confusing? You bet. But no researchers outside of his own lab could reproduce his work. That's not science. Every paper on CWD starts with, and I quote, "CWD is a fatal transmissible encephalopathy." Let me stress the fact that you do not transmit the disease. You transmit the infectious agent. My colleagues use sloppy language that illustrates their level of ignorance. As a result, their work has been sloppy, and the resource we love has suffered. If you believe that a normal host protein becomes infectious, regardless of the name you give it, show us the data. For those who need a refresher on 6th grade science, if a scientist truly discovered a new tiny life form that was able to multiply without nucleic acids, it would completely rewrite the books on biology. And as one skeptical science writer put it in the mid-1980s paper that has been scrubbed from the internet, and I quote, "If this prion thing were true, a redefinition of the meaning of life itself is needed." The fact is, no one in biology or medicine learned about this contradiction to the basics of biology in any courses on biology. I took them, no matter how advanced. I had to research this topic in depth outside of the academic knowledge that I heavily invested in to discover or attempt to discover why this was the case. More research dollars are being spent on CWD than any topic related to white-tailed deer ecology and management. But contrary to what many of my wildlife colleagues believe, there's very little productive research being done right now on CWD. Many millions of dollars spent, mostly falling short, looking at prevalence and susceptibility. You may quip, now wait a minute, what about the great research being done by researchers like Haley and Seabury and Henderson? While I would agree that some of this work is interesting and these are incredibly talented scientists, most, if not all, of it assumes that the prion theory has been cemented in the Scientific Hall of Fame, and more importantly, these studies are merely susceptibility studies. Variation in susceptibility is long since known. It's been known for scrapie in sheep, Sheep with a certain PRP are more resistant and have a longer incubation time. Great. Why not repeat some of the great work in the past that seeks to identify the true infectious agent? Why don't these researchers point out that the incredibly unethical and non-scientific approach that Prusner took with one goal in mind, to push a silly name for a cartoon drawing and win a Nobel Prize? They don't, because history has erased these facts. My message to these incredibly talented scientists with funding: what really needs to be done from a research perspective is identifying the true infectious agent and reproduce some very simple experiments done by scientists in the 60s, 70s, 80s. We do have reason to believe that the CWD infectious agent originated from scrapie-infected sheep. Scrapie is very similar to CWD in so many ways, and since the CWD science is failing to identify the true infectious agent, we follow the work of veterinarians and experimental neuropathologists in the scrapie, BSE, and CJD world. From here on, it's safe to assume that when I mention scrapie, I'm also referring to CWD. This podcast is supported in part by the Drop-Tine Seed Company and the Reload series of food plot seed blends that promote soil health, wild health, and diverse plantscapes that allow wildlife to thrive and maintain robust immune systems. The food plot you're gonna hunt in October and November is being built right now, 6 to 10 inches under the surface, by organisms most food plotters never think about. Soil microbes outnumber every other living thing in your plot by orders of magnitude. They eat the sugars and other goodies that plant roots leak as a result of photosynthesis. In return, they unlock minerals for plants, fix nitrogen, build organic matter, and protect plants from disease. That trade only happens when there are living roots in the ground. Bare and naked spring and summer plots are not resting. They're depleting. A diverse spring or summer reload plot is not just feeding deer, they're feeding the system that will feed your fall plot and fuel your hunt. This is the principle behind spring, summer, and fall reload. The reload seed blends from the Drop-Tine Seed Company were built specifically to keep that underground factory running through spring and into the summer-fall transition. Living roots in the ground, living plants on the surface capturing sunlight, microbes are fed, soil is getting better, not depleted. If you're trying to build a plot that performs in the fall and winter, lean on spring, summer, and fall reload to get you there. To learn more, visit droptineseed.com or visit the Drop-Tine Seed Company on Facebook and Instagram. Paul Harvey was one of the greatest radio broadcasters of all time. His signature and one-of-a-kind style and voice reached 24 million listeners each week. His massively popular and incredibly creative segment called The Rest of the Story uncovered and presented hidden facts and details of very well-known subjects, which ended in his famous line, made Paul Harvey an icon with everyday, hard-working Americans. For those of you who are too young to remember Paul Harvey, I insist you listen to some of his work. To those of you who, like me, just barely old enough to remember enjoying his approach to storytelling, his creative investigative journalism, and his iconic voice and tempo, I hope you enjoy this as much as I enjoyed creating it. So here we go. If you're interested in CWD from either side, I highly suggest you pay attention to what follows. Thank you for listening. Enjoy. You know what the mainstream CWD news is. In a minute, you're going to hear the rest of the story. I want you to meet Stanley, born in Des Moines, Iowa on May 28, 1942, exactly 84 years prior to the day this recording was released. Happy birthday, Stanley. Stanley's family later landed in Cincinnati, Ohio, where he attended junior and senior high, graduating from Walnut Hills High School. Although liking Cincinnati much better than Des Moines, young Stanley still found high school dreary and yearned for something new. In 1960, he set out for the University of Pennsylvania in Philadelphia. Stanley received his bachelor's degree in chemistry and later his MD from the University of Pennsylvania School of Medicine. In his own words, and looking back at that time period, he noted, and I quote, "I was cocky enough to think I could do research," end quote. If you know his peers and what they think of him, cocky is an understatement. During his fourth year of medical school, in the fall of 1967 and into February of 1968, Stanley conducted his research with scientist Ollie Lindbergh on brown fat cells. Also, in the fall of 1967, Stanley applied for a medical internship at the University of California at San Francisco, which helped him avoid being drafted and shipped off to Vietnam. After dodging the draft and a year in San Francisco, Stanley left California for Bethesda, Maryland, where he would serve as a research associate at the NIH, working with enzymes. It is his tenure at the NIH while working under biochemist Earl Statman that he credits as his, and I quote, primer course on the isolation and identification of the elusive prion, end quote. After 3 years at the NIH, Stanley sought to gain more experience by taking on an abbreviated residency in neurology before attacking his ultimate professional goal, his own laboratory. It was during this time that he experienced the sad struggles of a patient who appeared to have Creutzfeldt-Jakob disease, or CJD, known at the time as a slow virus disease, and justifiably so. It was that experience that forced Stanley to study up on the transmissible spongiform encephalopathies such as CJD, BSE, Kuru, and Scrapie. After discussing the case with the well-known scientists at the time, such as Gibbs, Gajushek, and Alper, Stanley had a hunch that turned the scientific world on its ear. A hunch, I repeat. No data, just a hunch. No virus, he said, no bacteria, just a rogue protein, a prion, he coined it, folding itself wrong and spreading like a bad habit. No DNA or RNA needed. How could this be? Nucleic acids, after all, are the genetic materials of life. They carry an organism's genetic instructions for the synthesis of its progeny. In other words, a new life form that really isn't alive, but can multiply without a genetic blueprint. Sounds like quackery. Bold as brass, that idea was. It flew in the face of everything we thought we knew about infection. The central dogma of biology itself was at question. But oh, how the accolades rolled in from the media and some of the more gullible science writers of the time. But what about Stanley's colleagues who had been studying these diseases since the 60s, many, many years before his arrival on the scene? Contrary to popular belief, the field was very quiet and calm until Stanley arrived. As one observant researcher and NIH Virologist Paul Brown noted, and I quote, "When Stanley arrived, if for no other reason than his prickly and aggressive nature, he leavened the field the way you leaven bread." End quote. The fact is, slow virus disease research hadn't seen much success from Gibbs and Gaddishek work to Stanley's arrival on the scene in 1974. Science writers from the time note that researchers were struggling to pin the actual infectious agent down. The truth be told, they still are today. After all, finding a virus of 20 to 25 nanometers in size in the brains of organisms is far worse than the proverbial needle in a haystack. It takes time, lots of time, and lots of something else that tends to be in short supply for all researchers: funding. You see, investigations like these are rather expensive. In fact, it takes millions of dollars to fund research like this, which means if just one man receives the lion's share of the available research dollars because he wines and dines with the money sources, it's not science at all, as we're at the mercy of the work, whether honest or not, of just one individual, one lab, one camp. That's not how science thrives. When Stanley approached the NIH for research dollars, they asked, and I quote, "Who the hell are you? You know something about enzyme chemistry, but you know nothing about virology and nothing about scrapie, and you never trained with anybody." End quote. In response, Stanley took a course on virology and later collaborated with William Hadlow, a pathologist at the National Institute of Health's Rocky Mountain Laboratories. Stanley and his colleagues went to work to isolate the scrapie agent until 1978, the year I was born, when the NIH opted to reorient slow virus research. They cut Stanley and his cronies off. Researcher Joe Gibbs noted that they desired new blood, and they ultimately forced Stanley and his colleagues to publish any data that they had. This turned out to be the catalyst of Stanley's desire to tell a Rocky Balboa-like storyline of struggles, hurdles, pitfalls, and major triumphs in his storybook career. In the months and years that followed, Stanley adopted some techniques used by British researchers in the 1960s. By 1981, Stanley claimed he'd purified the scrapie agent. He claimed in the absence of any proteins, they killed infectivity, suggesting that a protein must be the culprit. As a result, that no other researcher outside Stanley's lab or any of Stanley's co-workers could replicate. Regardless, Stanley immediately began brainstorming for the perfect name for his undiscovered discovery. The science publication called Discoverer quoted him as saying, quote, "Prion is a terrific word. It's snappy. It's easy to pronounce. People like it. It isn't easy to come up with a good word in biology. One hell of a lot of bad words people introduce get thrown away." End quote. Some of those who worked with Stanley noted that he seemed more focused on the perfect name than he was on interpreting the data itself. Stanley seemed to have a knack for public relations and rubbing elbows with the money sources and the popular, more influential media personnel. He leveraged the press to make him famous for discovering something that he hadn't found yet. The resulting fame brought the funding that would be required to outspend his opponents in the race to now actually find the real infectious agent. As a researcher seeking to dominate the funding, he even hired lobbying assistants. Over the years, he was said to have amassed $40 million in funding, leaving his colleagues to debate his Hollywood-like prion hypothesis on a shoestring budget. The world found out about Stanley's prion theory through an interesting but well-played series of events that no one talks about these days. While having lunch with a colleague, Curtis Morris, in the spring of 1981, Stanley claims that Morris encouraged him to summarize his data for an article in the publication Science. After all, the mantra of academicians is publish or perish. Knowing that the science paper would be a great place to introduce a new name for the brand new pathogen he claims to have discovered, Stanley got to work, and in June, he submitted his paper along with a note reminding the then editor, Philip Abelson, and I quote, that he needed to exercise caution in his choice of reviewers, since the scrapey field was polarized and he had already accumulated a cadre of critics. End quote. I bet that's not the type of independent, peer-reviewed process you imagined for a publication such as Science. So, to my fellow colleagues, if you're standing on peer-reviewed prion research from the 80s, you're standing on a house of cards. As the reviewers at Science vetted this manuscript, and just 2 months prior to the paper ultimately getting published in Science, on February 19, 1982, the San Francisco Chronicle was the first to release written word about Stanley's claim to fame in an article penned by David Perlman titled Tiny Life Form Found, which continued on the back page with the subtitle Weird New Form of Life. There's only one individual who would have leaked to the press, and Stanley has never denied it. In fact, he's written about it. The word prion became a household word among biologists without anyone ever actually reading the data, or lack thereof, in the publication Science. The result was that scientists and journalists alike repeated the prion hypothesis as though it was intensively vetted by an unbiased, peer-reviewed scientific journal. It wasn't. In theory, it was. In reality, the devil was in the details. As a result of the article in the San Francisco Chronicle, the press lifted Stanley up on their shoulders as he became famous through the lobbying of Stanley himself and his hired PR team. After making the press, Stanley proudly noted that, quote, the word prion became worldwide overnight, end quote. Mission accomplished. In 1982, Stanley's weak, overinterpreted data were published in the politically poisoned publication called Science. Science was slow to make their decision, with several of Stanley's colleagues and the reviewers of the publication questioning Stanley's claims. One colleague who reviewed the paper noted in a 3.5-page critique, quote, "Unfortunately, the style is critically flawed and is finally more suitable for a statement of personal opinion, even prejudice, than for a major balanced review in science." The reviewer continued on. Number one, the style is regrettably chauvinistic, not adequately reporting the work of prior and current investigations. And number two, the main concept that the spongiform encephalopathy agents are nucleic acid-free proteins depends mainly on the author's own, often unpublished and unconfirmed work. Regardless of pushback, Stanley's paper was published, and just like that, a made-up word cemented the prion hypothesis for decades to come. Research dollars slowed, and unconventional viruses disappeared. Only one lab continued to study infectivity in search for the true infectious agent. Interestingly, that lab showed that when you destroy all forms of PRP, the normal host protein, infectivity is maintained, proving that the misfolded protein is not the infectious agent. But recall that money talks in research. Without millions of dollars, researchers struggle to investigate the truth. Ironically, some new data in December of 2025 seemed to revive the infectivity research that was done 40 to 50 years ago. And it appears infectivity is present when the prion proteins are not. Instead, some researchers believe bacterial endotoxins are the culprit from contaminated feed. Whether time-tested or not, multiple hypotheses like these are necessary for science to science on. In truth, biologically, the infectious agent sure behaves like a virus. What about the word prion, pronounced pre-on and not prion? Recall, Stanley was in desperate need of a terrific and snappy word to describe the discovery that he had not yet found. In his own memoir, Stanley had asked, quote, "Why had I never taken an advanced course in word derivation?" Heck, after hearing how he derived the word prion, one could argue that Stanley could teach a course in advanced word derivation. The science writers at the time commonly reported that Stanley came up with the word prion because it was short for proteinaceous infectious particles, but not according to Stanley. As the story goes per Stanley, Stanley's journey in the name game started by identifying the most important words that describe the scrapie agent. As he saw it, protein, infectious, and agent. At first stab, he jotted down a P for protein, an I for infectious, and an A for agent. Adding an F, he spelled piaf. Stanley didn't like the way it rolled off the tongue. Plus, piaf apparently means sparrow in French. Back at the drawing board, he reports that he tossed the word agent out of his name game since it was non-specific, leaving protein and infectious. The story continues. One day, while eating lunch, tuna fish, lettuce, and tomato on mustard-drenched rye, to be exact, he notes that he, and I quote, constructed a two-dimensional matrix with infectious along the horizontal axis and the word protein on the vertical axis. The resulting word was prion. Then he flipped the O and the I to produce prion. If this story is news to you, sit tight because there's so much more you've missed. As the press raved on, Stanley's co-workers began leaking the truth. Paul Bentheim, a postdoc with Stanley at the time, revealed that, quote, Stanley rammed the word prion down the throats of everybody in the laboratory, end quote. Dave Bolton, who spent 3 years in Stanley's lab, confirmed Stanley's odd focus on branding the word prion. Both researchers then went to work for the competition. Bolton and Bentheim noted that Stanley coined the word prion as much to raise money for his research as for scientific reasons. Bolton and Bentheim also reported that Stanley hired fundraising experts, a luxury never afforded to other money-seeking academics, notably Stanley's opposition. The hired experts were charged with convincing private foundations and the public that his research shall be accepted as the final word. Bolton states, and I quote, Stan discussed this with us. He said, look, this whole area is getting lost in the muddle of slow virus this and unconventional that, and a whole bunch of other things. If we coin a new term for it and go out and tell people of the potential link to Alzheimer's, we're going to draw people's attention to this, and wait for it, and we're going to get money, end quote. By the way, I'm sad to report that we're making as much headway in Alzheimer's science as we are in CWD. What does this tell you? One of Stanley's researchers notes that the whole thing is so infuriating from scientific standpoint. The interpretation of the data has gotten so far from the data, it's hard to believe how it could have happened. End quote. These are the same data that my colleagues accept as the modern-day scientific dogma. What's behind Stanley's hard push to publish overinterpreted data and cram a made-up word, prion, down the throats of everyone, including his own colleagues and his own lab mates? Stanley's colleagues began to see the writing on the wall. You see, Stanley had worked so hard to do everything under his power to win a Nobel Prize. And since Gajdusek had already won the Nobel Prize in the slow virus field, Stanley knew he had to develop a different plan, away from the idea that the infectious agent was a slow virus. To support his motive, we lean on Frank Maziarz, a biochemist, and more importantly, Stanley's second in command. That's right, his right-hand man between 1978 and 1982. Maziarz quit after Stanley decided to publish the famous 1982 prion article. Quote, we were co-writing that, notes Maziarz. He continues, and I quote, I wanted it to be a critical overview in terms of the possibilities for the structure of the agent. By creating the name prion, he clearly wanted to push the entire interpretation in the direction of a protein-only agent. I said, there's no point in creating a name for something that we don't even know exists yet. But Stan's ego is unusually important in the way he interprets the data. He tends to jump to conclusions to give less credence to the fact that would discount his preferred interpretation of the results. End quote. Stanley's critics have repeatedly contested his sloppy science. However, these colleagues and critics are fighting an uphill battle, and they weren't even aware at the time of all of the politics going on in Stanley's own laboratory. It was as if the decision was made by those with the research money. It was as if the Nobel Prize was engraved with Stanley's name already on it. In 1997, the Nobel Committee handed Stanley the Nobel Prize in Physiology or Medicine, hailing his discovery as the key to unlocking these deadly enigmas. Many researchers, knowing that the infectious agent of scrapie and other TSEs like CWD had yet to be found, knew that a Nobel Prize would most certainly seal the deal for never-ending funding for Stanley, leaving nothing for those who felt confident that the prion theory was a hoax. Yet, even as the confetti fell, the grumbling started. Critics hollered from the rooftops. "Where's the proof?" they demanded. No one's ever cooked up a pure prion in a lab and watched it infect a healthy animal all on its lonesome. Here's a line I'd like you to use with the experts and just step back and admire their reaction. If recombinant PRP is not infectious, what data do you lean on to report that the prion is the infectious agent? What if these prions are just the sidekick, a scaffold, the symptom, not the villain? Maybe a hidden virus was pulling the strings, they argued, with prions acting as the doorman. One expert likened it to, and I quote, "the cold fusion of infectious disease." End quote. Flashy, appealing, but deserving a heap of scrutiny before we all buy in. Some researchers came down hard on Stanley, likening him to, are you ready for this? Biologists in the 1930s who had wrongly claimed that viruses were only proteins. I'll wrap up this incredible story with a poem written about Stan by one of his colleagues. There once was a young Turk named Stan who embarked on a devious plan. If I simply rename it, I'm sure I can claim it, said Stan as he pondered his scam. Eureka! Cried Stan. I have found it. Well, maybe not actually found it, but I talked to the press of the slow virus mess and invented a name to confound it. The fact is, there are many other incidences where well-respected researchers like Alzheimer's expert George Glenner and researcher Pat Mertz publicly criticized Stan for going to the press much too fast and even disagreeing with Stan's desire to release his opinions prior to developing the necessary data to support it. So, my fellow sportsmen, you are correct. There's a very good chance that the prion protein is not the infectious agent. However, researchers have given up and failed to investigate this extremely important piece of the puzzle. What is the true infectious agent? And why aren't we still searching for it? Many of my colleagues repeat sloppy information about CWD. they still stand on the mountain that states the infectious agent is only a protein. They've failed to pick up on Stanley's changing ways as he states, quote, "I never said it's only an infectious protein," end quote. Why haven't the experts picked up on this? And why does all of the research start by assuming that the infectious agent is a rogue, misfolded protein? It's because they're sloppy, and questioning is dead. Bolton summarizes this whole debacle by saying, quote, "Even if the scrapey agent was a virus and all the data were staring him in the face, my bet would be that he'd never see it," end quote. And so it is. The questionable work of Dr. Stanley Prusiner has been cited as fact, cemented forever in the scientific dogma to never be questioned by scientists who've lost the ancient art of questioning. Now you know why CWD researchers are failing to discover anything of true value. Is Alzheimer's research in the same boat because we're barking up the wrong tree? Now, you know, in part, why state agencies' flawed policies and management attempts to slow the virus are falling flat, and why their misguided hunting regulations pertaining to CWD are so unpopular with license buying, Pittman Robertson funding sportsmen. And now you know the rest of the story. Good day. The prion hypothesis does not fit the facts. If you invest a significant amount of time on the CWD topic from an independent position, you will learn that no one has ever proven that an infectious protein, which one fame-seeking individual calls a prion through some childish name game, does not exist. It hasn't been proven. It exists in the figment of somebody's imagination. Because that individual was given a Nobel Prize, so many don't question it. The truth is, they've given some really bad Nobel Prizes. They can draw a fibril however they want to draw it. The fact is, if they take a real molecule that fits the drawing and inject it into an animal, it is not infectious. Their prion drawings don't show real strains or real infection. Recombinant PRP that's folded and misfolded 10 different directions is not infectious. Most of my colleagues, the experts, are telling you it is because they haven't looked at the data, and they're assuming that the data that they hang their hats on are correct. They cite the Prusiner paper in Science, but they don't question it. Ask them if they've read it. If they have, they don't understand it. And truthfully, it's fiction, so it makes sense that they don't comprehend it. Don't take my word for it. Listen to many of Prusiner's own lab researchers who went separate ways after the paper was published in Science. The Prusiner camp excludes the citation of previously published peer-reviewed research from their papers. They exclude this previous work from anyone who contradicts their prion narrative. I can assure you, this is not how we were taught science. I'm here to tell you the science is not sciencing. The fact is, all of those scientific journals have changed. They're not about true science anymore. They're all about selling their publications, and they're not doing well financially as times change. When I was a student, you would see very few retractions or editors later publishing a note that says, "Hey, that paper we published last year, the results we described didn't actually come out that way. And older scientists who I talk to tell me that retractions in their time were practically unheard of. Retractions in the last 20 years have gone up exponentially, and nobody's paying attention. When a paper is published, the media runs away with it. When data or even entire papers are retracted, no one hears about it. The inaccurately reported results are already cemented into fact. What about the claim that there are no nucleic acids in the infectious agent? Nucleic acids are necessary for infection. Why are there nucleic acids in infectious fractions? Read the data. When you destroy nucleic acids but do not alter PRP, you lose 3 logs of infectivity. In other words, you only have about 1/1000th of the sample, or said another way, you have 1 part in 1,000 left of infection. They're misleading you when they say that the prion has no nucleic acids. I've watched my colleagues stumble here. I would ask, how do you define a prion? Per the prion camp, it's a misfolded protein. It doesn't have to have nucleic acid, but it also doesn't replicate and it doesn't create the infection. Could we be seeing a response to the infection? There's a good chance. We also have more data that show when you destroy all forms of PRP, infectivity is maintained. I repeat, when you destroy all of PRP, you don't destroy infectivity. This suggests that misfolded PRP or PRP amyloid is not the infectious agent. Certainly isn't acting alone. Is the normal host protein or PRP involved? Sure. Seems like it is. It's there. But that doesn't mean it's the infectious agent. I challenge today's scientists to repeat these very simple studies. Let's make sure we're on the right path before we continue making policies rooted in assumptions and the blind leading the blind. With this podcast, we've just begun to scratch the surface of this conversation. We created a new YouTube channel for the Drop-Tine podcast to release more information and discussions about CWD and other key topics that you don't hear about. We'll even discuss what guests are declining to speak and which prospective guests will only come on the show if we provide all of the questions that we plan to ask in advance. Not only is that not science, I consider it unethical and a disservice to my listeners. In the meantime, feel free to send me an email if you have any questions or any comments. Jason@droptinepodcast.com. Thank you for listening.